Welcome to today's lesson on genetic engineering as a form of biotechnology to modify organisms for food and medicine. We will explore how genetic engineering is applied to create genetically modified bacteria that produce insulin and genetically modified crops for higher yields and improved nutritional value. Our lesson today covers several key points. We will first recap the basic principles of genetic engineering as a biotechnological tool. Then, we will discuss how bacteria can be genetically engineered to produce insulin, which is essential for managing diabetes. Following that, we will look at how crops can be genetically modified to increase their yield or enhance their nutritional content, addressing issues related to food security. We have covered the process of genetic engineering in previous lessons. Here, we will look at practical applications of genetic engineering to improve medicine and food security. By applying biotechnology, we can tackle real-world problems more effectively, such as producing vital medicines and improving agricultural productivity to support growing global populations. Today, we are going to explore how genetic engineering can be used to modify organisms for the production of essential medicines and improved food crops. Genetic engineering is a powerful tool in biotechnology that allows scientists to transfer useful genes from one organism to another. Let's delve into two key applications: the production of insulin using bacteria and the development of genetically modified GM crops. Diabetes is a condition where the body cannot produce enough insulin, a hormone necessary for regulating blood sugar levels. Before genetic engineering, insulin was extracted from the pancreases of pigs and cattle. But this method was not efficient and had the risk of causing allergic reactions. Now, bacteria can be genetically modified to produce the same insulin as that naturally found in humans. The process begins with removing a small, circular piece of DNA known as a plasmid from a bacterium. Plasmids are independent of the bacterial chromosomal DNA and can replicate on their own. Scientists then isolate the gene that codes for human insulin from human DNA. This is achieved using special enzymes called restriction enzymes. These enzymes act like molecular scissors, cutting the DNA at specific points to isolate the insulin gene. Importantly, the cuts leave ends with exposed bases called sticky ends. The plasmid from the bacterium is cut open using the same restriction enzymes, resulting in matching sticky ends on the plasmid. The isolated human insulin gene is then inserted into the open plasmid. Another enzyme, DNA ligase, is added to join the sticky ends of the insulin gene and the plasmid together, creating a piece of recombinant DNA. This means two different types of DNA, bacterial and human, have been combined. The recombinant DNA plasmid is inserted back into the bacterium. The bacterium takes up this modified plasmid and incorporates it into its cellular machinery. The genetically modified bacteria are grown in large vats, where they divide rapidly. Each new bacterium carries the plasmid containing the human insulin gene and produces insulin as part of its normal metabolism. Finally, the insulin produced by these bacteria is harvested and purified to be used as a treatment for people with diabetes. This process has revolutionized diabetes treatment, providing a reliable and consistent source of human insulin. Genetic engineering is also used to modify crops to improve their yield, nutritional value, and resistance to pests and adverse environmental conditions. These modifications help ensure food security and provide essential nutrients to populations. Scientists first identify the gene that imparts a desired trait, such as pest resistance or enhanced nutritional value, from another organism. This gene is then introduced into the plant's cells. This can be done using various methods, such as inserting the gene into a plant virus, or using a gene gun to shoot tiny DNA-coated particles into plant cells. Cells that successfully take up the new gene are selected and grown into full plants. These plants now express the desired trait. For example, golden rice is a genetically modified variety of rice that contains a gene allowing it to produce beta-carotene, a precursor to vitamin A. This modification aims to address vitamin A deficiency in countries where rice is a staple food. There are many benefits of GM crops. Crops can be modified to produce substances that repel or kill pests. Genes can be introduced to help crops survive in harsh conditions, such as drought or flooding. Crops can further be engineered to contain more vitamins and minerals, improving their nutritional profile. In today's lesson, we discussed the role of genetic engineering in biotechnology, focusing on its applications in food production and medicine. We began by understanding that genetic engineering involves modifying the DNA of organisms to introduce new traits or enhance existing ones. This technology allows scientists to create organisms with desirable characteristics that can benefit humans in various ways. We explored the use of genetic engineering in bacteria to produce insulin, a vital hormone for managing diabetes. By inserting the gene responsible for insulin production into bacterial DNA, these microorganisms can be transformed into miniature factories that produce insulin efficiently and cost-effectively. This breakthrough has revolutionized diabetes treatment, providing a reliable and affordable source of insulin for patients worldwide. We then examined how genetic engineering is applied to crops to enhance yield and nutritional value. By introducing specific genes, scientists can develop crops that grow faster, resist pests and diseases, and tolerate harsh environmental conditions. These genetically modified GM crops can also be engineered to contain higher levels of essential nutrients, addressing issues of malnutrition and food security. This knowledge will be essential as we continue to explore the broader implications of biotechnology in the context of ecology and food production.